I'm very excited to talk about the NKT cell allogeneic cell therapy for the treatment of hematological and solid tumors. I am Dan Lang. I'm the president of Athenex Cell Therapy. So Athenex actually has been around since 2003. We aspire to bring innovative products from discovery to development all the way to commercialization for the benefit of cancer patients. We actually got our first drug approved back in 2020. This was a drug called Kuur for the treatment of actinic keratosis, which is a precancerous condition for skin cancer. And we recently made a big investment by acquiring Kuur Therapeutics back in May of 2021, which brings to us a very exciting allogeneic NKT cell platform that we're advancing in the pipeline. NKT cell is a very unique subset of T cells that go through a typical maturation in the thymus. At the same time, it also acquires very interesting and unique NK-like properties. For example, it could kill cancer cells very rapidly without priming. It has a very rapid release of cytokines, as well as being able to home into cancer cells very well. It also has NK-like activating and inhibitory receptors. And it only represents about 1% of the peripheral circulation. NKT cells have a very unique TCR receptor, contrasting the typical conventional alpha-beta TCRs that recognizes a peptide presented by the APCs, which are restricted by the HLA class 1 and class 2. NKT cells have what's called an invariant Tcr. They recognize, instead of peptides, glycolipids that are restricted by CD1d. Because the CD1d is monomorphic, it makes it a perfect candidate to be used allogeneically because it won't recognize foreign cells or other immune cells as foreign. We believe that NKT cells have a lot of very favorable properties that compare very well against other immune cells like T cells, NK cells, and gamma delta cells. It's been described in the literature for a long time that it bridges the innate immunity and the adaptive immunity by activating NK cells, T cells, dendritic cells, and really propagate and amplify the host's own immune system. As I said before, because it has an invariant TCR and the monomorphic CD1d, there's really no need to gene edit the TCR in order to use these cells allogeneically. It has very low to no graft versus host disease risk. These NKT cells could also kill tumor-associated macrophages and myeloid-derived suppressor cells, which turn the tumor microenvironment from a cold microenvironment to a more immunogenic environment that allow these cells to work better. These cells could also home into the tissues very well. They love to be in tissues, lymph nodes, and the tumor environment. These cells proliferate very well, very similar to the T cells after activation. And in humans, these cells could be resident for years, and it has immunological memory properties. This is a Kaplan-Meier curve showing basically stage 4 neuroblastoma patients. These are pediatric patients separating into two groups. The first group on the top here is those patients that have the tumor that shows NKT cells infiltration. And the bottom line is the group that does not have the NKT cell present in the tumor. As you can appreciate, the pointer is not working. The 5-year survival is actually very, very, much higher in the group that has NKT cell infiltration. I think it's somewhere between 60% compared to those that don't have NKT cells in the tumor of around 30%. This is a kind of indirect evidence to show that NKT cells could be very important in the anti-tumor surveillance of the patients. So we take these NKT cells and then we do genetic engineering on them to make them stronger and persist longer. We put in a CAR very similar to other approaches so they can target the tumor with a very specific antigen. We also insert the IL-15 gene so they can be secreted and make these NKT cells more potent as well as persisting longer. Lastly, we insert shRNA to knock down the HLA class 1 and class 2 to reduce the immunogenicity of these cells so they can persist longer and reduce the host versus graft effect that a lot of allo cells face. Here's our program. We have two programs in the clinic, 501 and 502, and then we have one preclinical program, Kuur 503. Our first program is an autologous approach. We're going, this is a first in man, using the NKT cells autologously, targeting a tumor that is widely expressed in neuroblastoma called GD2. After getting very safe and interesting data, we advanced the program into 502, which is an allogeneic approach targeting CD19 in CD19 positive lymphoma and leukemia. Here is the trial design for GeneKit-2, which is our autologous GD2 CAR NKT study that is currently being studied at Baylor College of Medicine. Our principal investigator is Dr. Hexy. This is a very standard phase 1 trial design because it's an autologous approach. We do the typical kind of ex vivo manufacturing as well as transduction with our genetic construct. Then we give the patients lymphodepletion with cyclophosphamide and fludarabine. And then we have a very, kind of starting at a very low dose. We have a 3x3 dose escalation at 3 times 10 to the 6, going about half a log at each dose cohort. We do the typical PK studies as well as biopsies when available of the tumor, as well as bone marrow. And then we do the typical standard imaging studies to look for any efficacy signals. Here are some flow cytometry studies that show at all dose cohorts we saw expansion of NKT cells. And then at the highest dose cohort, we saw the highest levels of NKT cells. And these cells persist for up to 4 weeks in our studies. On the left side of the graph here, these are flow cytometry studies that show before infusion at day -4, there is very little NKT cells present in the peripheral blood. However, by week 3, we saw a nice expansion of NKT cells. And what's really also good to see is that majority of these NKT cells in the peripheral blood also contain the CAR targeting GD2. As I said before, these NKT cells love to home in onto the tissue. So we did biopsies both in the tumor as well as the bone marrow. And, as you can see here, the pointer is not working, but at the upper right corner of the flow cytometry, you can see there is an expansion of the NKT cells. And it's nice to see that 86% of these NKT cells in the tumor contain the CAR, and really supports our hypothesis that these NKT cells have a really unique ability to home in onto the cancer cells. And the bottom part of the chart here shows a week 4 bone marrow biopsy, which again shows expansion of the NKT cells. And some of the NKT cells are CAR positive because there are other endogenous expansion of NKT cells. Here's a summary of the clinical data so far. We are at cohort dose level 4. We have enrolled about 11 patients with 1 PR and 1 CR and 4 stable disease in our hands so far. For the patient number 2, we actually give We gave 2 doses. Thank you. After we got the PR from the initial infusion, we gave another dose to hopefully drive deeper response and convert that patient to a CR. But the patient did not respond. However, patient number 10 here, after the initial observation of a partial response, we gave a second dose, and we were able to convert that patient from a PR to a CR. Patient number 3, we also saw anti-tumor activity, but it didn't qualify as a PR by the international criteria of Curie score. It's important to note that the safety of the product is very well tolerated. We only saw 1 grade 2 CRS. There was no ICANS, no neurotoxicity, and we saw the typical grade 3 to grade 4 cytopenias that are related to the lymphodepletion preconditioning regimen. Here are a couple imaging studies for those patients that responded to the treatment. The first patient here, patient number 2, had a pretty significant sternal mass before infusion, which reduced in size at week 4. And there is still a little bit of a residual lesion at week 8, and it didn't quite qualify as a complete response. For patient number 10, we saw a complete response. Here is the NIBG radioactive scan that showed lesions both in the thorax as well as the abdomen. And after 8 weeks, we saw complete resolution of those tumor lesions. After getting very promising results from GeneKit-2, which is an autologous approach, we went on to develop NKT cells in an allogeneic setting targeting CD19 relapse refractory lymphoma and leukemia. Unlike the first trial, GeneKit-2, that require ex vivo manufacturing, these cells came from the healthy donor. So it's a standard off-the-shelf therapy. The patient population, these are very relapse refractory. Many of these, a couple of these patients have received many rounds of prior chemotherapies, as well as first-generation CAR Ts. We started at a very low dose of 1 × 10⁷ per meter square, and we did a standard kind of a phase 1 safety study, looking at PK, imaging, as well as doing biopsies when they're available. Here are the initial results that we have disclosed so far back in May when we did the acquisition. So out of the 2 patients that have been enrolled so far, at the lowest dose at 1 × 10⁷ per meter square, out of the 2 patients, we saw 2 responses, 1 PR and 1 CR. And that's actually quite encouraging because we are at the lowest dose, and that's about 15 to 20 million cells, and that's a fraction of the current first-generation CAR Ts, which are on average 200 to 300 million cells. It's also important to note that we saw no graft versus host disease, which was something to worry about for an allo product, no CRS, and no ICANS. And we saw the typical grade 3 to grade 4 cytopenias related to lymphodepletion. Here's a graph of the normal B cell recovery, which I think suggests that by week 4, a lot of the allo NKT cells might have been clear from the blood circulation, or they have now infiltrated into the tumor. Here is imaging studies of the patient number 2, which got a complete response. This is a 58-year-old male with diffuse large cell lymphoma. He failed 2 prior rounds of therapies, as well as failing the autologous stem cell transplant. Prior to the infusion, patient had a thoracic mass, as well as a mass down in the pelvis, which reduced in size week 4 after infusion. What's really interesting is by week 16, this patient continued to respond and resulted in a complete response. So there was a conversion from PR to a CR, without any other intervening treatment in those 12 weeks, even though we couldn't see any NKT cells in the peripheral blood. Here is a flow cytometry study that shows in the lymph node at week 5, we saw an NKT cell expansion. And there are two populations that we discovered. One is the donor-derived NKT cells, which we know is A2 positive. And these cells contain the CAR, against CD19. We also saw a oligoclonal expansion of certain NKT cells that are CAR negative, and that represents to us, these are the patient's own NKT cells that might have been stimulated by the allo NKT cells that we infused. So in conclusion, we were able to effectively manufacture GD2 and CD19 CAR NKT cells to give this clinically that meet the GMP standards in very early days, but for the initial autologous GD2 CAR NKT program, out of 11 patients, we felt that it's been well tolerated. There is evidence of in vivo expansion of these in the peripheral blood, as well as homing and localization of these NKT cells to the tumor cells or tumor sites. We saw tumor regression in 3 patients with 1 durable complete response that went out to as far as 5 months. In the allogeneic CD19 anchor study, so far we have 2 patients. Out of those 2 patients, so far, it's been well tolerated. There is no evidence of expansion in the peripheral blood at the lowest dose cohort. Again, it's between 15 to 20 million cells. But we were able to show, homing of these NKT cells, at least in 1 patient in the tumor site. It's gratifying to see 2 out of 2 responses. We hope to replicate that kind of a data with more patients enrolled. And we're looking to advance our preclinical program targeting GPC3 for liver cancer sometime next year. I'd like to thank my team at Athenex, as well as our colleagues at Baylor College of Medicine. This is really a team effort without which there's no way that we can develop and advance this potentially very innovative, transformative therapy to help benefit patients. Thank you.